Okay, so I'm going to be talking about a project which, I hate to say it because it says a lot about my age, Sylvia's age, but I'm going to be talking about a project that's been going on at Iowa State between the USDA and Iowa State for almost 30 years. Is that correct, Sylvia? Over 30 years. And so we've been really lucky that we've had the opportunity to combine bioinformatics, molecular biologists, breeding, to start to understand what is happening to soybeans as they suffer from iron deficiency chlorosis. So iron deficiency chlorosis is a nutrient disease that's caused by a lack of iron. While there's lots of iron in the soil, it's actually unusable in most of the upper Midwestern United States. And so what IDC does is it causes interveinal chlorosis of the leaves, which results in reduced growth and end of season yield loss. So when we score IDC, we actually score it on a scale of 1 to 5, where 1 is a pretty healthy plant and 5 is a severely diseased plant. And the scale is important because every point on this scale, even going from 1 to 2, where you might not even notice it in the field, corresponds to a 20% reduction in yield. And so in 2004, it was estimated that 25% of the soybean crop in the upper Midwestern United States was lost to IDC. And even though farmers know that IDC is important, they actually prefer to use IDC susceptible lines because they yield higher in non-IDC conditions. So we are interested in figuring out what's the link between resistance to IDC and the effect in yield. So in our group, with Jamie and my group, we do a lot of work in hydroponics. And so for this first experiment, what we did was to grow the genotype Clark, which is iron deficient, in hydroponics for 9 days where they had sufficient iron. And then we transferred the plants to either iron sufficient conditions or iron deficient conditions. And we harvested the tissue 30, 60, and 120 minutes after the start of iron stress. We then collected the leaves and roots and then used these samples for RNA-seq. So what really surprised us is how fast Clark responds to iron stress. We identified over 3,000 differentially expressed genes in the leaves and over 6,000 differentially expressed genes in the roots. And what was interesting to us was that the roots respond right away within 30 minutes, and then the gene expression drops off. And then the leaves respond slowly, and they hit their maximum expression at about 120 minutes. So this is a really fast response. And with over 10,000 differentially expressed genes, how do you start to make sense of the data, and how do you analyze such a big data set? So one of the things that we use to make our life easier is to cluster the data by expression pattern. And so we took all of the genes from leaves and all of the genes from roots, and we clustered them so that all genes that went up at 30 minutes, down at 60 minutes, and up again at 120 minutes would all group together. And so one of the things that you can see is that gene expression is changing really quickly. Some of these genes that are down at 30 minutes are already turning on at 60 minutes, and then they're back off again at 120 minutes. And so these are very dynamic changes where we see differences in how fast they respond, and that there's very little overlap between leaves and roots, and even between 30 to 60 minutes in the same tissue. So since we could cluster these into clear expression clusters, we wanted to know if these clusters had biological function. So we looked at gene ontology biological process, and what we could see is that each of these clusters could be assigned a particular function. So for example, if we look at cluster 1, which is on at 60 minutes and off at 120 minutes, all of the genes in these clusters were associated with regulating gene silencing and the cell cycle. Cluster 2, which is down here, which turned on at 120 minutes, was associated with transport. Cluster 3, which is on at 30 and 120 minutes, is associated with iron transport. And then cluster 4 was associated with photosynthesis. So when we looked at the roots, it actually broke down into 2 main expression clusters, technically 4, but you can see that clusters 2 and 1 are very similar in their expression pattern, and that they're both on at 30 and 60 minutes, and clusters 3 and 4 are off. So when we looked at clusters 1 and 2, which were being turned on, we saw the classic iron response, where we have known transcription factors known to be involved in iron uptake in Arabidopsis. We see a lot of defense genes, being turned on very early, and then clusters 3 and 4 were associated with cell wall biogenesis and photosynthesis. So given that there's such a distinct response and such a distinct biological process is being affected, we wanted to know if we could start to tie transcription factors to these different clusters of genes. So this is just a plot showing all the transcription factors that we looked at. We identified over 50 different transcription factor families. On the left, I have the function of each of the clusters. And you can see, again, the dynamic response where leaves are slower to respond to iron stress and roots respond much faster. But if you look at these different clusters, you can see different families of transcription factors being activated. So now we can tie a transcription factor to potentially a biological function. So, what was interesting to us is that everything that we were seeing is very different from what's known in the Arabidopsis literature. Arabidopsis, when it's under iron stress, it induces iron uptake genes. And in soybean, regardless of the expression platform that we use, regardless of the time point that we've used, we've looked as early as 30 minutes and as late as 40 days. Soybean has a quadfecta response, where we see iron uptake being regulated, defense, cell cycle, and gene silencing. So we went back to look at what we know about the models in Arabidopsis. So Vert et al. Did a study to show that iron, whether it was sufficiency or deficiency, was signaling occurred from the shoot to the root and also from locally in the root to induce iron uptake genes. And Emoto and Goto demonstrated that it was in fact low iron that was responsible for the signaling. And then Stein and Waters have done studies looking at the expression of these iron uptake genes, and they used different Arabidopsis ecotypes that differed in the speed of their response. And so in the fastest ecotype that they used, Fro2 expression really doesn't spike till about 36 hours. And we see Fro expression at 30 minutes in soybean. So soybean is responding to iron stress incredibly quickly. So, since we had this model of what is known in Arabidopsis, we wanted to ask, does that model still hold true for soybean? So what we did is to look at all of the gene ontology terms in leaves and roots and look at their expression in both tissues at all time points. So when you look at these significant leaf go terms, what you can see is they're not on at all at 30 minutes, 60 minutes they're starting to turn on, and 120 minutes they're fully on. Whereas in the roots, they actually are activated at 30 minutes and start to turn off. When we look at the root go terms, we see the same pattern, where they're on at 30 minutes in the roots, and it takes quite a bit for their activation to occur in the leaves. So we think the model for soybean is that there's actually a quadfecta of responses, but the signal is going from the root to the shoot, which is very different from what we see in Arabidopsis. So this was really cool, and we were really excited about this. But this was just one nutrient stress that we were looking at. So Jamie's group has been looking at different nutrient stresses, and we really wanted to design our experiments so we could compare multiple nutrient stresses at one. We could take into account the age of the plant. We could compare how plants recover from iron stress, and we could see how they respond to a second stressful event. So these would be situations that you're much more likely to encounter in the field. And so basically, what Jamie did was to start plants all in sufficient conditions. Then one group went under iron stress, one group went under phosphate stress, and then the plants were allowed to recover by returning to nutrient-sufficient conditions. And then a second group got returned to iron stress or returned to phosphate stress. And then the fourth and fifth groups got a late either iron or phosphate stress, so that we were controlling for all of these conditions. And then she collected tissue from both roots and leaves. So what's the overlap between iron and phosphate? And what surprised us was that in the early time points where we were looking at the primary stress response and the recovery response, there's not a whole lot of overlap. The response to iron and the response to phosphate is actually quite different. But when you look at the second stress event where they had a second exposure to iron and phosphate, or at a late stress, suddenly the overlap between iron and phosphate becomes much more similar. So we wanted to understand exactly how, this just didn't really make sense to us. So what we decided to do was to actually visualize the GO data to figure out what was the difference between phosphate and iron stress. So if you look here, that iron actually responds at time 1, which was the initial stress response, whereas phosphate actually responds during recovery. And so what we think for all of these different GO categories is plants respond to iron deficiency, but they respond to phosphate sufficiency. So we see a very different pattern of expression. But what's interesting is that when we combine all of the time points across all of this data, we see the same soybean quadfecta, where the same classes of genes are being affected: photosynthesis, cell cycling and gene silencing, defense, and response to iron and phosphate starvation. So to kind of have a better idea of the transcription factors that might be regulating this, we also looked at the different transcription factor families, and this is again just the root data. And so what you can see is, in these first 2 time points, which is stress 1 and the recovery, iron and phosphate are not at all similar. But then with stress 1 and the second stress response and the late stress, suddenly the same transcription factor families are being activated. So to us, what this means is that recognition of iron deficiency occurs using different mechanisms than recognition of phosphate sufficiency, but they actually eventually use the same pathways. So, one of the things that expression studies have been criticized for is that expression studies have not really correlated well with QTLs. And when we look at our own data, what we see in soybean does not necessarily correlate with Arabidopsis. And so a lot of work has been done to characterize the main IDC QTL on soybean chromosome 3. So that was originally mapped in 1997, and then this has been successively narrowed down to a 250 kb interval. And within this interval, there was 2 known beta helix loop helix transcription factors, which according to the literature in Arabidopsis regulate iron uptake. And so these were identified as the candidate genes. And so we've actually tried to silence these genes using multiple constructs, multiple occasions, and we actually never see a phenotype in Clark. So you would expect if we had silenced these genes, we should affect iron uptake, and we don't see a response. And when we did RNA-seq of the plants, it confirmed that the silencing worked, that the targeted genes that were downstream of those were being silenced, but it suggests that these beta helix loop helix transcription factors were not the only causal gene. And this made a lot of sense with our expression data because soybean has that quadfecta response, where it is responding to iron stress, defense, gene silencing, and DNA replication. So based on everything that we knew, we had already predicted there had to be multiple genes based on expression data, and we also predicted that there would be multiple genes within that original IDC QTL. So we were really lucky to start collaborating with Danny Singh's lab, and they were using GWAS to look for different sources of iron deficiency tolerance. And so they had 2 years of field studies, 2 years, 2 replicates of hydroponic studies, and this was all done with the SNP chip. And on this QTL region on chromosome 3, which spanned 700 kb, they identified 16 significant SNPs associated across 3 or more experimental conditions. And so when we looked at this data, we were so excited because it was a non-random distribution of the SNPs. So we asked them to go back and look at the linkage across this QTL region. And it turns out that there's actually 4 different linkage blocks within the major IDC QTL. And so to us, that means that there is, in fact, 4 possible genes governing iron stress tolerance in soybean. And so based on what we know about the expression data, we can go through this interval and identify all of the different candidate genes. And when we identify these candidate genes, we're thinking about the soybean quadfecta: defense, DNA replication, iron uptake. And from these regions, we've identified a number of candidate genes that we're now characterizing using VIGs. So we're really excited about this research, and it's something that we're going to keep going after. So I think in conclusion, I think the thing that we've learned is that, you know, we work in soybean. Soybean has been under selection for 10,000 years. And in that selection, when we were selecting for improved yield, we were also probably selecting for the speed of the response. And so when you compare to Arabidopsis, which has not had the same selection pressure, you might find that there's actually a different model. Secondly, you know, I didn't really talk about the rest of the GWAS study that was done in Danny's lab, but there is data to suggest there are other regions in the genome that contribute to IDC tolerance. And then finally, you know, I want to thank all of our collaborators, because this was such a group effort where you had to combine breeders, computational biologists, molecular biologists, and none of this would have been possible, you know, without people being open-minded and willing to look at other data that maybe made them a little bit uncomfortable. So with that, I'd like to thank 30 years of collaborators and also Jamie, who has done a lot of the work as well.
Any questions?
Bob.
I should probably know the answer. So my first question is, do we know if there's a yield drag associated with that QTL on chromosome 3? Is that ever been looked at closely?
I don't know that it has.
I'm
So,
just curious because you were talking about the conclusions about selection and whether or not there's
yeah.
sort of a balance.
Well, and so I don't know if you remember a long time ago, we looked at that GM RPA3 gene, and RPA3 is in the middle of that other IDC QTL. So when we looked at that region, there's an RPA subunit in that IDC QTL, and when we silenced that, the original gene, it caused like really tiny plants. Jashali?
Did you find any expressed gene for IDC exposed to other abiotic stress such as drought or
when we looked at that QTL region, did we find genes that were associated with other stress responses? Yes. And I think, so what we did to characterize that locus is we took into account all of our expression data. We looked through the literature to see if those genes had ever been associated with other stress or abiotic or biotic stresses. We also looked at Arabidopsis just because a large, you know, there are functions for some of those genes, and that's how we picked our candidates. So yes, some of them had multiple functions. Mm-hmm.
So silencing doesn't cause phenotype, but one of your experiments, right?
Yeah.
Silencing. So do you think that maybe there is enough amount of protein when there is suppression to take up
When we did the silencing, because we kept getting failed, like a failed phenotype is no phenotype, right? So that was why we did RNA-seq to confirm that we were getting silencing, and we also confirmed that the downstream genes of that target were also being silenced, and so they were, just that in itself was not enough to cause a phenotype.
Yeah, perhaps there are so many things that take up metals, and there are so many transporters that can transport so many metals.
Yes. And it could be that you need, since there's so many, appears to be so many genes in that QTL, that having just one is not going to give you a phenotype, that we would have to start putting them together into multiple VIGS constructs.
So any of the overexpression?
We have not tried overexpression.
Michelle, how long do you think we are on your work to try to answer the question that he was asking about yield relationship? You know, like taking that region with all the genes that we have there, and then put that region into some susceptible IDC cultivar.
I think the hard part is that since there appears to be a yield drag, and it's tightly linked to that region, is that you have to break the good with the bad. So you have to think about it in like, so the plant clearly stops growing when it's under iron stress.
Yeah.
And the reason it stops growing is because it's like, ooh, you know, let's take all of our nutrients and put them to this response. And so you don't necessarily want to break that response. You want to tweak that response. And so I think that we probably would want to use like genome editing to change some of the expression of some of those genes rather than using a straight classical approach. Because I think if you put that whole section in without breaking it, it's going to be another line that is great in IDC conditions but not in.
So maybe I didn't understand that right, but what, biologically speaking, what is a phosphate sufficiency signal? So, because deficiency is easy to understand, but sufficiency, at least from in terms of your experiment, seems like you made it deficient and then added phosphate. That's sufficiency.
Yeah, that's sufficient.
That's not.
It's recovery to sufficiency, yes. And so I think that it must be a recognition of, you know, if they can recognize low nutrients, they also must be able to recognize high nutrients, because, you know, certain nutrients can be toxic at high enough levels. So I think it's, you know, kind of trying to figure out, like you say, I mean, we don't know what the mechanism is, you know, but it appears that that seems to be the difference.
I'm also wondering if, is it possible to think of it as a delayed recognition of an insufficiency compared to iron? So maybe it comes in much later and looks like.
So we clustered the data where we put into different Go biological terms, where different things. So for some categories of genes, it's clearly nothing happens till recovery. Though there are some categories of genes where it seems like it's almost ramping. So like that there are some genes that it's a slow response.
All right, let's take Michelle one more time.